all, this is Dr. Mobeen Sayed with one more episode of Long Story Short with Dr. Bean from FLCCC platform. Let's start our discussion. The topic today is brain fog and if there are possibilities to reduce that or improve that situation with lifestyle changes, some supplements, and some diets. Please realize that this is not a medical advice. This is more of an informational session plus a review of the latest studies about neurogenesis. And now, what is neurogenesis? Believe it or not, within our brain, there are structures where neurons, new neurons, are produced every day. That is what is neurogenesis. Genesis means production, neuro is neurons. Neurogenesis. I would have called it neo-neurogenesis, new neuron generation, but I think neurogenesis means a similar thing. The neurogenesis also tells us that in our brain, we also have neuronal stem cells that are the parent cell. A stem cell is defined as a cell that can make its own kind of cells, plus it can make cells that can become other cells. That is a stem cell. So we actually have neuronal stem cells within our brain that can give birth to new neurons. And a new neuron that is born has a complex set of processes and steps to become functional part of the brain society, the other neurons society. So the new neuron that is born, first it has to become differentiated. Differentiated means to become a cell of a special type, for example, to become a neuron. Then it has to develop all the features of a neuron. Then it has to become integrated in the current circuitry. Then it has to function there correctly. So there's a lot of responsibility for those newly born cells and the cells around them to help them become mature and integrate. Let's look at all of this. Of course, the topic means, today's topic, that if there is any probability, any reason for the neurogenesis to reduce, then we'll have a problem. And if there are ways to improve neurogenesis, then we would have an improvement. That is the basic crux of the discussion. Let's start. Here, I'm going to show you the links first. So this is FLCCC. You can find the other lectures over here as well. Now, this is a very good discussion on TED by Dr. Sandrine Thuret. She is talking about neurogenesis over here. There are some studies here about neurogenesis going as far back as 1970s, but then as recent as June and May of this year, 2022. So here are some studies. I will refer to some of those studies in the discussion today as well. So with this, now let's look at the drawings. So first, within our brain, this part of the brain, which is somewhere in this area, is called temporal lobe. Deep in the temporal lobe, there is a dentate gyrus. There is an area called dentate gyrus. Within the dentate gyrus, there is a piece of brain tissue called hippocampus. So today we are especially talking about hippocampus because a lot of neurogenesis goes on in hippocampus. And there may be a question in your mind, what is the benefit of making those new cells? There are two parts of the answers. One, this also means production of new cells also mean older cells are dying. And production of new cells means these may be in the areas which are responsible for new things happening, new events. So new memory formation, new pattern recognition, new event recordings. For all of those, we can actually give birth to new neurons and ask them to remember this. I'm simplifying it. And at the same time, older neurons could be dying as well. So hippocampus is a very important part of our brain for this critical function. Hippocampus is also part of the limbic system. Limbic system is our primitive brain, if you will. It is responsible, limbic system, with the emotion, with the sex, with the olfaction, with the basic functions of our survival and our species propagation. Now, hippocampus is tasked with helping with memory. It is a very plastic and vulnerable part of the brain. What does that mean? Plasticity means there are new cells that are being produced. And those cells can learn and do new things, new functions, new patterns, new memories. At the same time, it is vulnerable because if there is any event that is going to reduce the production of new cells, that would cause hippocampal damage. Because the older cells are dying, new cells are not being produced, hippocampal function is now going to become impacted. That would impact almost the whole brain's function. So it's a very, very vulnerable area. For example, chemotherapy. Some patients develop depression with chemotherapy. Even after the chemotherapy, they have depression. Why is that? One of the cause is that what do we do with the chemotherapy? When we administer chemotherapy, most of the time what we are trying to do is we are trying to reduce the production of new cells, because cancer is really uncontrolled production of new rogue cells. So what we do is with chemotherapy, we just stop all kind of new cell production. That would also stop, these drugs would also stop production of new neurons in the brain as well. That means hippocampal neuroblasts or new neurons will not be generated there. And one of the symptoms of that is depression. And it is interesting that when we give anti-anxiety drugs, these drugs, many of them actually increase the production of new neurons in hippocampus and bring the person out of depression. Now you might ask this question that, hey, all right, fine. During the chemo, there are less neurons being produced, and so there is depression. What happens after the chemo? Why there still is a persistent depression? And the reason for that is that when the chemo suppresses the cell production, the newer cells are not produced right away with the full number one. Number two, there is a gap. So let's say the chemo was able to suppress the new cell production for 15 days. Now 15 days, we did not produce new 700 neurons. When the chemo is stopped, the newer neurons are going to now start being produced at a lower rate, but they still have to produce the neurons for all those missed days. That is why depression continues for some time even after chemo. Then hippocampus is also responsible for emotional responses and organization of emotions. It is also responsible to help with recollection of the past experiences. So when you say, all right, I can remember this or I can remember that, that is the recollection of the past events, and hippocampus helps with that. And it helps with imagining future. So imagine any issue with the hippocampal area would not only make the person feel that they are not able to recollect and recall the older memories, but they would also not be able to project in the future. And they would come back and they'll say, I have brain fog. I cannot do the normal cognitive functions correctly. In the hippocampus, there are 700 new neurons produced every day. The reduction in new cell production in hippocampal area causes depression. If the cell count is less, then earliest and most severely affected structure in neuropsychiatric disorders is hippocampus. So whenever there is any reason, for example, there is chemotherapy being given, or there is inflammatory state, or there is some other, let's say folic acid deficiency, which is folic acid is necessary for cell replication, or let's say vitamin B12 deficiency, or other material that is necessary for cell replication, or enzymes, for example, zinc or copper, if they are less, then a early symptom will be the effect on the hippocampus causing depression. So depression sometimes is not just a psychiatric problem, but it could actually be a physiological problem where the new neurons are not being produced correctly, or even if they are produced, they're not being matured and integrated correctly. So this just tells you how important it is for us to make sure that neurogenesis continues to occur in the brain. Now, hippocampus is also very important for spatial navigation. So this is very important. If I know I'm sitting here and I'm doing this work and there is the camera and here is the mic and here is the recording system. Similarly, you know about your environment and you can navigate that. That is the spatial navigation. And hippocampus helps with that as well. Hippocampus helps with the pattern recognition. This is a mouse. This is a pen. This is a computer. This is a mic. Pattern recognition. Imagine if I lose the capability of recognizing pattern. We are nothing but pattern recognizing machines. So as soon as I cannot identify things correctly or fast enough, I'll say I have brain fog. Hippocampus is very important for encoding new memories. So when you have an event and it is going to become a memory, we have to not only form the memory, we have to label the memory as well. Is it a good memory or bad memory? Is it associated with the smell or food or event or whatever? That encoding of the memory is also with the help of hippocampus. And imagine any impact to hippocampus almost is going to be a devastation for the patient. Now, interestingly, hippocampus can be influenced by hypothalamus and it can influence hypothalamus. Now, you know from our other discussions that hypothalamus is a big boss for the endocrine management, hormonal systems management. And hippocampus is helping hypothalamus. So when there is a problem with hippocampus, then hypothalamic control on the suprarenal glands is not correct. The result is an increased amount of cortisol. So a patient whose diet may not be correct, or who may be on chemo, or who may be in an inflammatory state, who may have folic acid deficiency or B12 deficiency, and they're depressed, it may be that they also would have increased cortisol levels, which would exacerbate the whole situation even more. So as a physician, keep an eye on this. Depression plus increased cortisol level may indicate that there is something going on with hippocampus. Now, abnormality of the cells or the number of cells and the integration of the cells in hippocampal area is linked to epilepsy, depression, Alzheimer's, Parkinson's, strokes. Early parts will be brain fog, early indications to the patient. Patient would not know. They would not be able to come in and say, I have a problem with my hippocampus. They would say, I cannot think about the memories. I cannot plan about the future. I cannot recognize things fast enough. I cannot name them very correctly. I cannot understand where I am. That all can be an indicator of a problem with the hippocampal neurogenesis. So this all is to say, how do we do neurogenesis or how do we maintain it? So let's continue to see some more background to this. I think that this background is not shared or presented anywhere out there. I watched a lot of videos, read a lot of topics. Every topic or a video addresses one area, but they do not create this foundation. So I think it is a useful foundation to have. So in the hippocampal area, we have neuronal stem cells or NSC, neuronal stem cells. Interestingly, these neuronal stem cells, so imagine with me, in the hippocampus, there are some cells sitting there. They can make new neurons. They are parent cells. Some glutamatergic signals, glutamate if released in that area, signals these cells to start making new neurons. Now, what triggers glutamatergic signals can be a lot of factors within the brain's own function and the body's state. So the first thing, glutamatergic signals to the hippocampal stem cells cause the stem cells to start proliferating. Proliferation means to increase in number. Hypothalamic supramammillary nucleus or SUM is also involved in this. So remember we talked about hypothalamus, that is another structure. Above the hypothalamus, there is this supramammillary nucleus. So within the hypothalamus, there is a nucleus called supramammillary nucleus. That nucleus, when receives the glutamatergic neurons or signals, then this supramammillary nucleus or SUM as we call it, it further starts regulating the neuroblast activity in hippocampus. So neuron stem cell regulation is through the hypothalamus, and hypothalamus or one of the nucleus of the hypothalamus, which in turn is regulated by glutamate. So in simple terms, glutamate finally influences production of the new neurons in the hippocampus. Now, we can make new neurons, just like we can make new babies, but then there is a responsibility of maturing them. You cannot just make a baby and leave them alone and let them be on their own selves and mature by themselves. So once those baby neurons are made, we have to nurture them or guide them to become neurons, to develop their little neuron connections, and then to integrate and function. So the story is not over yet that we have new neurons. Once the new neurons are made, then we need a different kind of signal, and that is GABAergic signals or GABA releasing signals. The story started with glutamate. Now we have GABA. GABA and the signals that are causing the neurons to release GABA in this area will now cause the maturity of these neurons. So as neuroblasts mature, neuroblasts are those new little neurons, baby neurons. As they mature, GABAergic inputs from supramammillary nucleus of hypothalamus help regulate dendrite production. So on a baby neuron, there are many filaments coming out of it. Those filaments, those areas where it receives input are called dendrites. And those where it sends output, the wires through which it sends the output are called axons. There is normally just one axon and many dendrites. So dendrite production on this baby neuron, imagine growing hair on the baby neuron, is only possible if there are continuously available GABAergic signals. Why am I talking about them? If there is a disruption of glutamatergic signals or GABAergic signals, then we would have a problem with producing new neurons. And for this discussion, we have to accept that there is a possibility of producing new neurons. And I'll discuss that a little more down the road in this lecture. So dendrite production is done. Then the GABAergic input is still needed for these neurons to integrate in the society or society of the other neurons. So if I can now share it as a diagram, here are the stem cells sitting in hippocampus. The glutamatergic signals went to supramammillary nuclei of the hypothalamus, which in turn triggered the stem cells to say, make more cells. And the stem cells would renew themselves, plus they'll make more copies of them, which is called proliferation. This all started with glutamatergic signals. Once the proliferation has occurred, now we want these new cells, these new baby cells, to become neurons. That would be called differentiation and maturation and integration. So now we need GABAergic signals. So GABAergic signals would start the differentiation of making this new baby neuron into a more evolved neuron. We still need more GABAergic signals to have this neuron start producing the dendrites. Dendrites are from where it would receive the signals. And then we still need more GABAergic influence for these new baby neurons to start getting connected with the other neurons sitting in that area, in that society, and become integrated. If any of these steps fails, then we may have neurogenesis attempted but failed, right? So if glutamatergic signals are there, then the neurogenesis would start. But if GABAergic signals are not there, then these newborn baby neurons will die. Or if the GABAergic neuron signals are there, but they're not sufficient, then this baby neuron might develop some dendrites and not integrate correctly. So delicate, complex, and continuous process, which needs a lot of continuous balance within the brain of various signals. And it is so easy to imbalance that or create an imbalance just by inflammatory states, chemotherapies, B12 deficiency, folic acid deficiency, and many other. And I'm going to discuss them. This is the background to say, how do we improve this situation? So now the question is, how do we accomplish neurogenesis to continue? Remember, whenever there is a sign of depression, there may be a cause of problems with the neurogenesis. We also know inflammatory states can cause disruption of neurogenesis. We also know that chemotherapies can cause disruption of neurogenesis. Any reason, any factor that is involved in cell replication and growth, an absence or disruption of that factor will lead to disruption of neurogenesis. And one sign of that is going to be depression. So now, how do we make it better? First, there are some studies that are done on mice. There are some studies that are on humans. There is a big controversy in this field, and that is that maybe this happens in mice, or maybe this happens in macaques or primates, but not in humans. So I would show you studies which are on both sides. There are some studies that say it happens in humans. There are some studies that say it doesn't happen in humans. I'm going to show you all of them. So first thing is enriched environment. Now, what does that mean? I'm going to show you enriched environment right now. So I'm going to go back to my main screen and get ready. This environment is now enriched for you. Why? It is novel. Why? This is not how it was a few minutes ago. That surprise, you liked it or you did not like it, is a different thing. But that surprise, that novelty that you felt actually causes neurogenesis. So they saw that in the mice studies, those mice that had a boring environment around that didn't have those little running wheels or didn't have the tunnels and the stairs, these mice did not have sufficient neurogenesis. And as soon as they gave them the little wheels or the tunnels and other things and mice started running around, they started having neurogenesis. So first thing, an easy thing to do, create an environment around you or get into an environment that would surprise you, that would be novel. Go to another place, go to a club, go to a Starbucks, go outside, go to a forest, go to something new where your brain would say, oh man, this is a new set of signals for me. And it needs to produce new memories for that. It needs to produce new emotions with that. It needs to encode that whole new thing. For that, it needs to produce new neurons as well. It's not that the memories can only be built with the new neurons, but new neurons are important for all of these new things that you just experienced. So we all become used to, we may have a beautiful environment that somebody else comes into that and says, wow, this is amazing. But for your own self, it may have become a routine, a boring thing. You see it every day. So put yourself in a situation where you change that, you create novelty for yourself. So take a detour while walking or driving, surprise detour. Go somewhere where you have never taken a turn before. Travel. Look at the beautiful or messy, unique situations. It's not necessary. It always has to be beautiful too. Even if you saw a messy situation, you would have this new event for yourself. Have a new fragrance. Smell new fragrance. Plant new flowers. They would create new visual and olfactory sensation. Have a new taste. Art, art, art. I keep asking everyone to indulge themselves in art. As we age, one way to keep ourselves sharp is art. And it's not necessary that you have to become master in arts. You just have to attempt art. And that process of doing an effort makes your brain develop new neurons and stay sharp. Now, before I continue with more factors, I want to address a controversy. The controversy in this field is, as I just said, that some people say that this doesn't happen in humans and some say it does. So here are some studies, and in the links, all such studies are linked, or a lot more, not all. So first, here is a large study on primates. So this study, if you look at the time here, published 30 May 2022. The intent of the authors was that if we can prove that neurogenesis is occurring in primates, then we humans, being very close to primates, will have neurogenesis as well. So in this study, they actually said, look at the robust adult neurogenesis in the primate hippocampus. Overall, our data set demonstrated robust neurogenesis in the adult macaque hippocampus, revealing the presence of all key neurogenic precursor cell populations, including RGLs, IPCs, and neuroblasts. Our analysis also identified substantial differences in neurogenic processes between rodent and non-human primates, such as different marker gene expression pattern and an augmented proportion of immature granule cells in primates. So they said it's not necessary that we have to find the same markers that were present in mouse in the humans as well, because the markers are different. That is why somebody who's looking for the same marker that they found in the mouse and they're looking for that marker in the humans, that's why they're failing. So they said, at least in the macaques, which are very close to humans, there is a lot of neurogenesis occurring, but the markers are different. So this is one study. Then here, this is 6th January. So that was May. This is 6th January this year. Single nucleus sequencing finds no adult hippocampal neurogenesis in humans. So this is January of this year, 2022. And they're saying we didn't find any evidence of neurogenesis. You saw this in May, where they're saying we found evidence or we can extrapolate evidence. Now, if I continue, here is one more. This is an older one, Nature. If you look at it, this is 1998. And here they say, using immunofluorescence labeling for BRDU, and that is here, and for one of the neuronal markers, NEUN, CAL binding and neuron specific enclose, we demonstrate that new neurons as defined by these markers are generated from dividing progenitor cells in the dentate gyrus of the adult humans, where the hippocampus is, Our results further indicate that the human hippocampus retains its ability to generate neurons throughout life. So while this discussion continues, there is another set of researchers who are doing something very interesting. They're saying, fine, happening or not happening, why not we try to influence the neurogenesis and see if the patient improves? And they're seeing improvement. So there is a clinical evidence. So we'll continue with the clinical evidence and the results of that. So one, you saw novel environment. Stress negatively affects hippocampal neurogenesis. So these are again studies, non-human studies, that when the stress occurs in animals or macaques or mice, the production of new neurons reduces. So there is a study that I have the link for, and I just showed that in the beginning. There is a study where they said that if neurogenesis is affected by stress, and the younger adolescent humans are for sure having neurogenesis, even if we are not, any stress on them in their adolescent stage of life can create lifelong effect by reducing the number of new neurons produced, integration of those neurons, and the robustness of hippocampal area. And that deficiency can continue forever. That means, and nowadays with the COVID and with the vaccines, the neurological issues has become a lot, and youngsters need even more protection. Then running increases cell proliferation and neurogenesis in mouse dentate gyrus. They give them the wheels or they give them tunnels and stuff, and all of a sudden there's a blast of neurogenesis. And it is understandable that when we do something, whatever, it causes us to remember it. There is an event that needs to be processed. We need to judge that to say, is it a good thing or a bad thing? If you ran near a flower, you have to remember that smell and say, wow, I smelled that nice fragrance, and so on. So there is a lot of events that would become memories, and that would trigger neurogenesis. Now, what are other factors that can cause neurogenesis or disrupt neurogenesis? So on this side are the factors that can help produce new neurons. On this side are the factors that can disrupt new neuron production. So as I said before, novelty, fun, anything that you have to remember, anything remarkable. Maybe this lecture, and you say, in this lecture, Mubin had turned the lights off. And just to remember that, hey, this was a lecture number 15. He had turned the lights off. Your brain will have to make some memories, and it would trigger neurogenesis as well. Reading. So once again, it is not the act of reading, but it is the information from reading that needs to be retained and need to create new connections and memories. And remember, when we make a new memory or whenever we have a new event, it is integrated into the rest. And that is what reading would do. Good sleep. They have seen that the sleep deficiency causes reduction in neurogenesis. So good sleep is very important. Sex. Sex also causes neurogenesis. So that, again, according to Dr. Sandrine, too much sex that causes a person not to sleep well would have a counterproductive effect because of the sleep deprivation. But one can adjust the time for sex, but still, sex is an important factor. Learning new things, which may not be by reading. You may be doing something with hand or art or playing or running or listening or seeing. So all of those are learning new things, which would once again trigger the brain to say, learn this and understand this and memorize this and connect it to the rest of the things. Hippocampus has to work with that. Neurogenesis would occur. Intermittent fasting. Can you imagine intermittent fasting? So we did this talk a few weeks ago. Intermittent fasting causes the older cells to die faster and newer cells to be produced. So intermittent fasting triggers neurogenesis. What a beautiful thing. Caloric restriction of 20 to 30%, just 20 to 30% of restriction from your normal habit triggers neurogenesis. Blueberries trigger neurogenesis. Flavonoids, for example, dark chocolates, trigger neurogenesis. Curcumin, and Dr. Paul Merrick, we had a call a few days ago and he said, remember, curcumin needs to be mixed with fats to be absorbed correctly. That is why it is mixed with oils or milk. It is put in food which has oil or mixed with milk. So curcumin in a good absorbable state triggers neurogenesis. Caffeine, resveratrol, which is present in many kind of nuts and stuff and also in the red wine. Omega-3 fatty acids. Once again, many nuts and those things plus salmon, fatty fish. Folic acid, vitamin B12 especially, or vitamin Bs. Why are these here? Because they take part in the help to produce new cells. Zinc, same thing. It is a cofactor for many enzymes that are working for the cellular divisions. Vitamin A, similar behavior. High sulfur diets. So those diets that are high in sulfur, many of them are vegetable diets or vegetarian diets, they have glutathione in them, which would in turn, glutamatergic signals will become robust with that. Then crunchy food, chewing food, fragrant things. Why? So if you are eating soft food, according to Dr. Sandrine, it doesn't cause too much neurogenesis. But if you're chewing or masticating, then it does. And I think it is very understandable. A soft food is almost like nothing in the mouth. But a crunchy food, something you're masticating, something that is making a sound, something that has a texture in your mouth or in your tongue, creates an event that needs to be processed and learned or rejected, or you said, wow, this is great, or you said, yuck, this is not good. So crunchy, masticating foods or foods with textures are very important. Any event that would improve the blood supply to the brain would also help, which also tells us that why the brain fog in folks with the cardiovascular issues or vascular inflammation and microclotting. Not only it is causing tinnitus and the other neurological symptoms, the depression and the brain fog. And that is a pandemic that is going on nowadays, at least with the COVID. Then on the negative side, stress severely impacts neurogenesis. Aging process causes neurogenesis reduction as well. It is a normal process. However, we can slow it down further by the other things I just described. Inflammatory state of the body, chemotherapy, high saturated fats, high sugar content, but I read in one study that fructose doesn't have that effect. High sugar content reduces neurogenesis, but fructose does not have that impact. Reduced vitamin B, A, and folic acid. These are necessary for the cell production. Alcohol, ethanol, soft diets. So this is the discussion for today, and we'll stop here. I hope that the takeaway from this is that especially youngsters are going to be more prone because they have a lot of neurogenesis for sure. For adults, we are prone nowadays because of COVIDs and vaccines to have neurogenesis impact, which would cause neuropsychiatric issues and depression-like outcomes. And some of these methods, lifestyle changes, diets, supplements can help or may help. So again, talk with your doctor, figure out what is the best for you, what can you be doing and what you should not be doing, and then go from there. Thank you very much for listening in, and I would see you next week. Bye for now.